Chemistry and Cell Biology Part 2. Take notes on Unit 1-1, pages 8 through 12. Nucleic acids. These are polymers or nucleotides, where a nucleotide is a sugar, phosphate, and nitrogenous base. What you're looking at there on the left is DNA. This is a double-stranded molecule, two DNA strands side by side held together by base pairing, A to T, G to C. You can see there circled a nitrogenous base or nucleotide with nitrogenous base, sugar, and phosphate indicated respectively. The DNA forms a double helix, and you can see differences with RNA and DNA. RNA is single-stranded, and DNA has deoxyribose with adenine, thymine, guanine, cytosine. RNA is single-stranded with the sugar ribose and has uracil. Now, DNA also is important in a different way than RNA is. DNA stores a genetic code in the nucleus. The double-stranded nature increases stability. Nuclear membranes protect the DNA from chemical breakdown, whereas RNA is really the working copies of DNA used to make proteins by translation. Messenger RNA, transfer RNA, and ribosomal RNA all have their functions there, and you should know their functions. Okay, chromosomes. We always hear chromosomes when we talk about DNA, but really chromosomes are about half DNA, half protein, and they're compacted. The only time you see chromosomes is when the cell's dividing. Humans have 23 pairs or 46 total chromosomes. There's two types, autosomes, which are the same in both sexes. There's 22 pairs of those, and there are sex chromosomes, in which is one pair, is XX in females, XY in males. Now, a special nucleotide that is one of the building blocks of DNA or RNA is adenosine triphosphate. This molecule is also used as an energy carrier and receives a lot of attention as being the energy carrier of the cell for all chemical reactions. The reason you can do that is the last two phosphate bonds that you see there circled are super high energy bonds, and when you break those bonds enzymatically, you can release that energy to go to different processes. So how does the ATP store and transfer energy? These high energy bonds, when they're broken and you form ADP and phosphate, the energy is transferred to things like muscle contractions, active transport, protein synthesis. Of course, anytime energy is transferred, the efficiency of that transfer is relatively low for most systems, and you lose a lot of energy as heat. And of course, as mammals, we transfer this energy into body heat to maintain our temperature at 37 degrees C. This slide is just to show a summary of cellular structures. We're going to now transition to the cell structure and just review that very quickly. But this picture is going to be available in Bolt. You can download this. It is not in your lecture guide. What I want to do is just kind of point out that cells that you see in these pictures are generalized and will have a lot of structures that any one cell may or may not have. As we take a look at the cell's organelles, let's first begin with cell membrane structure. And the cell membrane, of course, refers to really a plasma membrane. It consists of lipid, protein, carbohydrates. Those are the three main molecules that are found in the cell membrane. Lipids about 55% composition, protein 40%, and carbohydrate 5%. These things are not arranged randomly in the cell membrane. They're actually arranged in a very specific way for their function. There were several different models of how cell membranes were put together. Until about 1972, they put together something called the fluid mosaic model, where the phospholipids form a fluid layer that is mobile, is oil-like, kind of like a droplet of oil, and then proteins and cholesterol are moving about in this fluid layer, kind of like logs floating in a stream. The phospholipids prevent things generally that are not lipid soluble from getting inside the cell. So if we look at the functions, the lipids, phospholipids specifically, separate extracellular from intracellular fluid. The proteins associated with the phospholipids and provide functions of the membrane. Some of them are enzymes, some are transport proteins, some are antibodies and other immune proteins, and carbohydrates or sugars added to the protein or lipid to make them functional. A term I want you to be familiar with is sarcolemma, a term for the muscle cell membrane, which is a special term. Most organelles have at least one membrane around them, a plasma membrane, that separates the contents of the organelle from the cytoplasm. The nucleus is unique in that it has two plasma membranes around it, and this form is called the nuclear envelope. If one membrane is a great barrier, then two must be an impossible barrier. And to get things in and out of the nucleus, you do need nuclear pores. Nuclear pores are not just holes, but they're actually protein-guarded pores that regulate very carefully what goes in and out of the nucleus. Also, make note that the nucleus is typically the largest organelle, and that it stains darkly, typically, and is very visible. You'll often see a small dark spot on the nucleus called the nucleolus, where ribosomal RNA is made and ribosomes are made. An organelle that's closely related to the nucleus and often very close to it physically is the endoplasmic reticulum. It's a system of sacs and tubules containing a substance called endoplasm, special cytoplasm. There are two forms of it in structure. The rough ER sacs that have ribosomes attached, which is the little red dots that you see here, and then smooth ER that has tubules lacking attached ribosomes. These two types of ER function very differently. The rough ER synthesizes proteins in selected areas. The rough ER proteins go into lysosomes, cell membranes, and they can be secreted as proteins in the ECF. Example, that would be insulin. We also see that the rough ER can be enlarged in neurons, forms things called Nissl bodies. This is specialized rough ER that synthesizes neurotransmitters. And then the smooth ER has three major functions in lipid synthesis, detoxification, and calcium storage. The Golgi body is closely related to those structures. It is a series of flattened sacs. Its function basically is to modify proteins synthesized at the rough ER. It does this by adding monosaccharides and removing amino acids, so that, for example, a molecule called proinsulin is the inactive form of insulin. It is simply cut up into smaller pieces, has some of the amino acids removed to become an active form called insulin. Okay, ribosomes. This is the only organelle that does not contain a membrane around it. And actually it's kind of interesting because it's an organelle that bacteria also have. So I'm going to say bacteria lack organelles, they really don't. The ribosomes are composed of two small and large subunits. Function is that they come together, the two subunits come together, and they serve as a site of protein synthesis, and they can be located as free ribosomes in the intracellular fluid or cytoplasm and the rough ER. Another organelle that you readily recognize is mitochondrion or mitochondria plural. It's a double membrane organelle with two compartments. The innermost compartment is the matrix. There's the intermembrane space, which is the second compartment. The two membranes are the inner membrane, which is highly folded with the cristae on it, and increases surface area of the inner membrane by folding it. The outer membrane is not folded. In terms of functions, the inner membrane serves as a site for electron transport system. Matrix is a site for the enzymes of the Krebs cycle. These two things together serve aerobic respiration. Note that the mitochondria is about bacteria size. Lysosomes are often called the suicide sacs of the cell because, in terms of their structure, they're just membranous sacs, a plasma membrane surrounding hydrolytic enzymes. These hydrolytic enzymes, by their name, use water to break down large molecules. They can digest any molecule in the cell, and their function is intracellular digestion. They're very prevalent in phagocytic white blood cells that are going to engulf and digest bacteria. Another similar organelle, it looks very similar under the microscope, is something called a vacuole. It is also a membranous sac, just a plasma membrane that surrounds an inner fluid. That fluid is typically used as storage, and this can be fats, oils, which we typically will see in adipocytes. Otherwise, it can be storing water or ions, but that's not very typical in mammalian cells. Okay, cilia and flagella are structures that basically have microtubule arrangement and support from the cytoskeleton. Microtubules are proteins arranged to protrude from the cell. They're arranged in 9 doublets and 2 singlets. Difference between cilia and flagella then, and you can't tell them apart in cross-section, but you can tell them based on their arrangement on the surface of the cell. Cilia have many short projections, flagella usually 1 to 3 long projections. Ultimately, their function is for locomotion and movement to create currents within the body. The flagellum is found on sperm for locomotion. Cilia, we're going to see lots and lots of cilia when we study the respiratory epithelium, and you can see that it covers the surface of columnar cells there that then move trapped particles through the mucus layer to sites where they can be broken down and removed.